So we are going to go ahead and get started for this last section, which is, okay, perfect, we have enough chairs. Okay, so we have a Q&A with most of our presenters. I do want to do one quick wrap-up detail before we start this, just in case anybody has to leave early. It's very important that those of you, again, as I'm going to just reiterate, collecting CEUs, you have to fill out this survey, but those of you who are not collecting them, we still highly encourage you to fill this out. It's how we're gathering feedback, and we'll improve upon this course each year. So we really want to hear everybody's feedback. And all of the slides are now available in the shared folder, and recordings will be coming to the course website soon. My hope is sometime in the next month or so, but we do need to wait for video to be processed and get them online, so just be a little patient with us, especially during the summer months when we want to be outside and with our families as well, on top of everything else. So with that, I am going to make this go black for a little bit so we can focus on our lovely speakers. So for the Q&A, again, we want to hear from all of you, but I will kick off the questions, since a lot of people don't always want to start. So my question I will pick on 3 of you to answer this question, not everybody. We spent 2 full days talking about tools and processes, and for the busy clinicians in the audience who want their key 1 to 2 takeaway points from this course, quickly summarize what your key points for them and takeaway points for them would be. And because this is the first time I get to put Heidi on the spot and not vice versa, I'm going to start with Heidi.
Good question. Takeaway points. So I guess I would say that if you're a clinician or a genetic counselor and you're getting test results back from a laboratory, that even if you trust what the lab says in that report, knowledge evolves daily, and you should, always be a skeptic and continue to pursue further knowledge if the evidence isn't enormous in terms of the amount of data that supports that variant, and that you should pursue things like segregation studies in the family to confirm that variant segregates with disease in the family, not just test the unaffected as predictive. And you should continue to check ClinVar and ensure consistency between your patient's phenotype and the variants that have been identified. As well as always remember that there are other patients out there to benefit from the knowledge that you've gathered, and either only ordering tests from a lab that shares data, figuring out ways that you can contribute to these resources by providing phenotype both to the laboratory and to public knowledge bases. There are clinicians and other genetic counselors who are submitting data separately to ClinVar, not just the labs. And so you too can contribute in useful ways to enhance the value of the genetic information you provide to your patients. And you can also take a gene off a clinical report, and you can sign up for Gene Matcher and submit that gene in there for patient matching. So I think as clinicians out there, you should all recognize that you have a role in the data sharing world and ensuring that knowledge is up to date for your patients. That would be my takeaways.
All right. And since you are a clinician, what are your takeaways for other clinicians?
So I definitely really like to be looking up every variant I see, both in GENOMES and in ClinVar, so I also encourage people to do that. There's so many different databases we can look at, and I'm finding new ones all the time that are really interesting. So just keep paying attention to what you see on Twitter, what you hear about when people talk, that check out some of these. Like the Decipher one is something I've been using for 2 or 3 months now, which is obviously a site that's been around for a long time, but some of the new features are really great. The other thing is I've really tried to bring the families along with me. I work with families that have pediatric onset rare disease, which is different from some of the cancer predispositions. and other adult onset things. But the families are the parents are the ones who know their child and their child's condition the best, and encouraging them to be part of this data sharing process. There's things like MyGene2, where families can go on and share their information, both their phenotype, their genotype, or even their VCF, with others in the rare disease community to try to learn more and have these families be able to work together. So stay strong, keep at it. I know it's a lot out there.
And Erin, as the genetic counselor.
Yeah, so, one thing that it strikes me whenever I talk to other clinicians, particularly other genetic counselors, about things like variant interpretation, et cetera, is that we frequently hear, well, that's not my job, that's the lab's job. Why would I look into this? Why would I bother? I don't have time, I'm very busy. And I know that it seems overwhelming at first, especially when you're brand new to this. Perhaps, you were not taught how to do any of this in your genetic counseling programs. But I think that it's so critical to at least have a grasp of what the laboratories are considering when they are interpreting your variants, so that when you get that report yourself, you can take a look, an educated look at what they've got, and notice if something is missing or something doesn't seem to make sense to you. And then if you find something like that, having the confidence to call that lab up and ask questions. Say, why is this? Why are you saying this? Why are you saying that? I noticed this other lab has a different interpretation. Why is yours different? And kind of be that advocate for your patients that you've always been in the clinical setting, but maybe extending that out into their laboratory testing results as well. Having an understanding of how this information came together also helps you to better explain the results to your patients. So I don't think, people think, oh, well, it's doing double work. The lab already did that. Why would I redo it? It's not a redo. It's not saying that you don't trust your lab. It's to help you help your patients if you better understand what's going on in your lab report.
So that was my kickoff question. I expect now all of you to have questions as well. So I will open it up to the floor if any of you come forward. I know there are some in the audience, so I will wait patiently and then pick on people who I know might have questions. Mel, you have the floor.
I guess I was just wondering, and probably for a subset of kind of 5, 10 years from now, what do you think this course would be about? Or kind of what advances would you successfully have made and what the new challenges would be?
Daniel, I'll pick you.
Thank you, Sam. It's kind of incredible to think about what the resources that we're looking at now, like GENOMES, for instance, what they will actually look like in 5 years' time. I mean, there will obviously have, have multiple millions of individuals that have been sequenced. Of course, everyone will be incredibly keen on data sharing because Heidi and I and others have pushed them so hard to get their data out there, so that this will all be aggregated together into large databases. But most importantly, we'll also have, in a way that GENOMES doesn't currently have, linkage between that information and phenotype data. So you can imagine being in a situation where as a mutation arrives in a clinical setting, you have at your fingertips information both on, firstly, has that variant ever been seen before in population reference databases of millions of individuals? If so, how common is it across different populations, including very fine-grained population structure? And has it been associated with phenotypes in those populations? Secondly, as ClinVar continues to grow astronomically, you'll be able to see, here are exactly the patients where variants like this across this gene have been reported in a whole series of individuals with particular phenotypes, and again, fine-grained information about the specific syndromic phenotypes associated with those particular mutations. And then finally, and I think really critically, information from systematic functional assays, where we can actually say, even if a variant hasn't been seen before in a patient, we know that this particular mutation has been introduced into a well-calibrated functional assay for that particular gene, and that particular mutation does or does not actually have an effect on some important function of that specific marker. And so with that type of information, I think we start thinking about variant interpretation less as an art, which is what we're doing now, and more as a quantitative science. All that information makes it possible, I think, to make much more quantitative and probabilistic estimates about the likelihood of pathogenicity. So I guess what I'd love to see in this course in 5 years' time would be a discussion where we're no longer talking as much about the uncertainty and the caveats about the resources, but instead, how do we just bring all of this information together in a quantitative and statistically informed way to make pretty clear-cut decisions about whether or not a variant is actually pathogenic and causal in a particular patient?
So I agree with Daniel that I do hope in 5 years' time it is less about the binary, is it or isn't it pathogenic, and that we have a better understanding of those variants that are in fact definitively disease associated. But at that stage, we will be much more in the realm of sequencing healthy individuals. And then the question is, all right, what's the chance my patient is actually going to develop this disease for which they harbor a definitively pathogenic variant? And trying to better understand what are the both environmental and other genetic modifier factors that may lead one patient to develop a disease and another to not, and how can we really start to predict true risk, or even if we can't predict true risk, what kinds of approaches do we take as clinicians to mitigate risk and reduce it without necessarily certainty of the likelihood of development, so that we're taking sort of approaches that with an understanding of an element of risk, but recognizing that there will always be uncertainty. So I moving it really from is the variant, can it cause disease, to will it cause disease?
Anybody else want to answer? Or Melissa, yeah, go ahead. Should be on.
So yeah, I completely agree with both of you. I think that in 5 years, it's not just going to be about the genomic data. It's going to be about the phenomic data, and phenomics is on the rise. And I know I've been saying that for 10 years, but it's finally going to happen in 5 years from now. And I think that really speaks to what Heidi was just saying. It's going to be about using genotype and phenotype and environment data in a structured way together for precision health, not precision diagnostics, and that how do we actually help people live healthier lives based upon their genetic endowment and the environments that they have around them, and make good decisions about their health care rather than seeing them in the clinic when they're in crisis, right? So it'll be about how do we save people's lives a priori by having the tools and the algorithms that help us interpret this vast body of knowledge. The second thing I think is going to happen is also related to the data sharing, and patients are so much more empowered now to participate, and I'm so grateful for efforts like yours to really help them go along, and they're going to walk around with their own data in hand and want to understand it in the context of the greater ecosystem of data. And so anything we can all do now to help everyone share better, but especially the patients are becoming increasingly educated. And so I guess in 5 years, I'd love to see patients in this course and participating in these types of analyses themselves. So.
If you can use the mic, as I almost yelled it without using the mic. If you can just bring it down so when this is recorded, people can hear the questions. Thanks.
So one of the things that came up the last 2 days is really or maybe a big aim was to collaborate and to share data and the benefits. And it is very much needed and very much appreciated. And one of the methods that you put forward was standardization to help with these collaborations. And my question is, once that is established, how do you prevent the system from becoming too rigid to be maybe an obstacle to innovation, or to maybe exclude outlier hypotheses that might still be true?
So just so I understand your question, you're asking, do we lose granularity in some ways by too much standardization? Is that, and how can we ensure that that doesn't happen? Is that kind of your question?
If you give too much credit to standardization, then perhaps it could become an obstacle for thinking outside of the box.
Yeah.
And sometimes for rare disease, that will be required.
Yeah, I mean, it's a great question, and I think on the one hand, in order to do a lot of the work we want to do at scale, where we can actually make use of the numbers that we have of genomes, exomes, millions of patients with phenotypic data from electronic health records. We do have to standardize in some ways, but I think Matchmaker Exchange is a great example where at the end of the day, when you're taking those 2 or 3 or 4 incredibly rare cases, the only way you can figure out really how to harmonize those phenotypes is to start asking and sharing very detailed information. I've seen both a phenotype be sent by a set of HPO terms, and then I've seen a clinician write out a very careful, thoughtful history of that child or individual's development and manifestation and assumptions about what was a primary phenotype versus a secondary phenotype. And there's nuance there that is definitely not captured by a list of HPO terms. And so I think we all do need to appreciate that just standardizing everything as HPO terms is not, no offense, Melissa, is not going to always get us to the last mile, and that there will be things that to get those subtleties out and sometimes really understand the pathogenic mechanism of disease and what is the primary underlying issues at play, which might then inform treatment in other ways or ways to do natural history studies, et cetera. In some cases, we'll get down to the granularity that you just can't always capture entirely in a structured form. So I think the key thing is we don't ever want to lose that. We just want to try to get the bulk of analyses done so that we can take advantage of high throughput approaches and the scale that we are increasingly able to benefit from, but recognize that you will have to, pull out the paper and pencil at the end and, kind of look in depth, if that makes sense.
Yeah, maybe I could speak a little bit to that too. So so just to be clear, I would never advocate for removing free text from from any of us. I think we have to have Goldilocks solutions though, where you absolutely, and I think being able to have free text notes on every patient in a pedigree file, in anything that you're transmitting, it's a requirement. And actually, those are places where we mine for information. Those are really important places where people can write their findings and their thoughts. That whether it's clinical notes or protein structure interpretations anywhere. And so in all of the sort of data standards that we work on and that we try to develop, where we're actually promoting innovation at scale, because we've got that standardization, we never want to lose the ability to say what we think and be able to transfer. And that's kind of a human, a human thing that we all have to bring to the table, all of us, in all the contexts that we have. So.
So over the last 2 days, what we've been talking about is describing highly striking histories, so either de novo pediatric cases. What do you hope to see in the future that could help describe less penetrant syndromes, so for example, cancer syndromes? What can we provide to you as clinicians, or what is your dream database to do that?
So I mean, it's a great question. Actually, I don't know if Judy's still in the audience out there. No, not. Judy Garber and I were talking about, in fact, hereditary cancer and the challenge of family history, and in fact, the inaccuracy of information that's often provided. And so, there are some newer tools, particularly for proband of a family to be able to share that they have, been identified with a pathogenic variant for a cancer syndrome, for example, and want to be able to share that information with family members. And so there's these online apps that have been developed to help, send out. You can just put in your family members' email addresses. It will send notices out, allow them to go into an interface, enter their own clinical data and family and their own data, so that you essentially create a family history from each individual, as opposed to the proband trying to remember that Aunt Sally had some kind of cancer, because we all understand the inaccuracy of that. And so it is certainly my hope that we can use tools like this. So, and actually some really nice data came out of Emer Kenny, and some of the work they've been doing with EHR data, where they use certain proxies for relationships, and were able to connect families together via the other information. I forget exactly what sorts of things they were using, but it really created ways to get more accurate relationships and more accurate, and in fact they were using genotype data to actually connect people, so that you can actually take a real record as opposed to a conveyed family history. And so I think some of these types of approaches hopefully will give us better information to inform pathogenicity, but also better information to inform risk to an individual. Is this a variant that's in a family with 100% penetrance across the pedigree, or is it in a family where one person had disease and 5 others had the variant and never developed disease? And what ages were those when, in terms of their stage of life? And I think that will, help, and I deal with cardiomyopathy. It's a similar issue in terms of understanding true risk. And that was sort of what I was alluding to before as we switched to the question of, what's my true risk? What's the penetrance? James Ware actually presented some really nice data at the Keystone meeting a week ago, looking at Titin variants for dilated cardiomyopathy, and the likelihood of developing disease is correlated with alcohol intake, and just really beautiful data, showing the correlation between alcohol intake and the likelihood that you actually develop cardiomyopathy due to your Titin variant. So I think those, but to do those, you need really good data, good careful cardiac measurements, and good histories of alcohol intake. So I think we will need to continue to build more reliable resources of the type of information that will inform both pathogenicity and risk. That makes sense?
I think biobanks, we're really getting to the era where right now we're collecting, people are participating in these large-scale projects, but we don't have enough of the genotype and exome data that goes along with it. Geisinger is one of the leaders here, where they have been exome sequencing a large proportion of the Western Pennsylvania population. So they have this large group. They're mostly related, and like over 25% of them are related, and they're able to estimate penetrance for some of the cancer variant. They've been talking a lot of ASHG and other places. And so this is going to grow, because what you need is people who are willing to participate in research, funding for exome or genome sequencing, or at least a GWAS to start, and then connection with electronic health medical records that are codified in a way that they talk to each other, which is really hard in the United States. But in some countries like the Genomics England project, where people are in the national healthcare system and more of their records hopefully talk to each other. I actually don't know specifically, but I believe they do. And to have more of that codified, that's really what we're going to need to get that all together. And that's hard in the U.S. because people move more. They go to different medical systems. Their relatives are often in different systems than their children are. And so getting that detailed history across generations is much more challenging, but it's going to happen, especially as the costs of sequencing come down.
And I think that also just speaks to the point of really remembering that the patients are our partners here, and trying to think of creative ways to engage them. Using the medical record is a great option, but it's not always viable in certain situations, just like you described, like if people are moving around or if ones are not talking to each other. So that's one of the reasons I'm really excited about Genome Connect, just trying to get those people to actively agree to participate and to engage them over time. Because I think if we leverage that properly, we can also get more longitudinal data that we may not have had access to otherwise. I mean, these are people from all over the world that would never be collected together in the same EHR. So we can kind of look at it from both angles, and I think that that's a cool thing.
I heard you say a couple of things, and this might be seen as devil's advocate a little bit, but it's more of a concern and a question, and I don't have a solution. I heard you say people should communicate with their family members, provide email addresses, enter information about their alcohol intake, risks for cancer. So EMRs, I just think we're going to have to figure out a solution for people like me who really don't want to share. I'm all for good health and preventing cancer and talking to people about your risk for this and your risk for that. But when my 20-year-old cousin goes to Ancestry.com and puts in all this information because he doesn't care or know about privacy, he's giving my information too. Who's going to use the databases? Who's going to figure out? Who's going to hack? How are we going to protect people, especially when the thing that flagged was the alcohol intake, right? Someone's employer is going to look up on the website to see if they're at risk for this, and then they're going to come across, this is how many drinks I have a week. I mean, I know I'm making it a little bit more, I don't know what the adjective is, but I just think we need to be cognizant that flags are going to go up in people's heads that will make them more reluctant to share instead of more willing to share and anticipating some of these things. And some of them they really are issues that we might not be able to fix, but if we at least address them, maybe that would help. I said a lot, but I think you know what I mean. It's really about privacy and sharing and what you're revealing and what you don't want to reveal. And you all have the cousin that says, blah, 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 I don't want to know this, but you might save their life, but they might not care.
Yeah, and we're always going to deal with those issues. People should and only can share what they are comfortable sharing. And there are ways to do studies where you're getting data in aggregate. You're not sharing individual, this person had the alcohol intake and that. And there are also subtleties, drinking 1 glass a week. Most people are not concerned about sharing that level of information, but even things as, or 3 a week or whatever, you don't have to acknowledge that you're an alcoholic to have this information be useful. So I think, each individual needs to be comfortable sharing the level of information that they feel comfortable sharing. And we also, when we're doing studies, we have to be, and there's been a lot of thought, I can tell you, I've been involved in a number of meetings at NIH thinking about, and Daniel's been involved in these too, like the ability to even show aggregate statistics, which has been shut down at NIH for a long time because of any fear of figuring out that you're in that database and not even knowing which one of you is in there and that level of information. And there has been a distinction between sensitive information, psychiatric illness, and HIV status and other things that we all recognize can be very sensitive information, and sometimes treating that type of information differently than other information. Like, do I get exposed to the sun every day? Yeah, okay. So I think there, we had this conversation a little bit earlier. We can never guarantee that there'll never be any breach of information. Anybody who says they can is lying or doesn't understand. But it goes back to the risk-benefit question, right? And a combination of the comfort with which you understand the utility of the information you're sharing. And if I had a set of family members potentially at risk for a treatable disease with a very bad outcome, that I felt like I could help by conveying my information, that's a more compelling argument than, just, sign up on this website and share all your data, right? So the more we can convey, why is this useful? What is it gonna, what are we gonna learn from this information? How will this help either other individuals out there or direct family members and things like that? And that may sometimes change the level of information an individual shares and the environments they may share that information.
Yeah, those are all excellent points, and I guess there was multiple things wrapped up in that question. One was about the challenges about what happens how do you make sure that people know what they're doing when they share their own data, and that's a hard problem. It's obviously one in the Facebook era we're wrestling with all the time. It's been in the news a lot, and I think is at the top of people's minds. So there, it's really just about transparency. I think you made that point really clearly, and that is a matter of both presenting risks in a way that doesn't downplay the true risks, but at the same time doesn't overhype them. And I think one of the challenges with some of the conversations both Heidi and I have been engaged with is there is a tendency sometimes to go to backlash in the opposite direction and get very angsty about anyone sharing any data because of the potential imaginary harms down the line. And I think we do have to be careful about that too. So there I think transparency is going to be key. The second thing though, the interesting thing about genetic information that you alluded to was the fact that it's quite different in the sense that you sharing your own information explicitly provides information about your relatives in a way that they have never provided consent for. And this is a new type of privacy risk that society as a whole hasn't really ever had to wrestle with before. And we saw that in such an interesting way. I think when the serial killer was caught using these genealogical databases, such a clear demonstration of the fact that each of the people who signed up for those databases, the killer himself didn't sign up, but enough of his relatives had, incidentally, that they were able to triangulate his specific identity using that approach, which is kind of remarkable. And I think there's not an easy solution to that. There's this balance between personal autonomy, that is my right to be able to donate, to be able to contribute data or share my own data to a resource, as I think should be our fundamental right, but at the same time clashing with the fact that by doing so you impinge on the rights of your family members. And there's no easy solution to that beyond, I guess, like encouraging people to actually have conversations with their families, but you can't enforce that. So I think it's a challenge that society has yet to grapple with, but we need to solve it in the era of genetic information. Otherwise, there are things about data sharing that we just can't fix.
I struggle with data sharing, even like with Facebook or Twitter. When you take a photo and there's people in the background, do I have to get their permission? Like, what am I allowed to share? But I'd actually like to see there be more legal protection against identifying people who are participating in research studies. That we know this comes up that for the most part you can only really identify which person it is if you have some of their genetic material. But just in general, like, it shouldn't be okay for people who are volunteering to participate in things, for you to share their information. We can't share people's financial information. There's penalties against that. We are perfectly, as a community, able to create laws that have either fines or jail time even for sharing information that should be protected. As a society, we can decide those things.
Hi. So coming to the question of curating variants and trying to do them effectively and rapidly. In a clinical laboratory, we are often faced with the situation where we have to have high-quality traceable evidence, which is with clear audit trails, and with So when you're reading papers, two different scientists can read the same paper and, of course, evaluate it differently, and they both would have done it correctly in their own way. And now we have to do this faster. So, what is the future for machine learning and natural language processing tools? Because right now, I think NLP helps to identify that your variant is in the paper, but most of the variant level information is hidden in the supplemental tables, which currently NLP is not picking. But that being said, even if it's able to do that, the context in which the variant is presented in that paper is so prone to human interpretation, because I think machine learning and natural language processing are not at a point where they can understand the subtleties of human language, and scientific literature and clinical literature is just so full of subtlety of human language, especially the discussion sections of papers and all that. So where do you see machine language and natural language processing? When do you think they'll be able to make some inroads to help you rapidly curate excerpt evidence in different sections of your classification, be it a functional study, be it a functional meiotic transmission, or and such? So what is your outlook on that?
Yeah, I mean, to be honest, our whole system today is really quite crazy, that we all have lovely databases that we track all our information, and then the way we communicate with the rest of the world is we write a text-based paper with a bunch of figures that have to be converted to not even useful figures, but the journal requires you to convert them to some archaic thing that I can never manage to do myself. to then put it out there. And then we developed machine learning and natural language processing to try to take the data out of the paper that was originally structured to start with and bring it back into some usable form. It's actually kind of crazy, right? Now, there are increasing examples where the journals require that data to be accessible in structured form, submitted to dbGaP or sometimes more useful places, and with annotations on it so that that analysis can be replicated by other scientists. So we need to continue to push in that direction. I'm now part of a monthly phone call, because I don't have enough monthly phone calls, with journal editors discussing requirement that all variants and papers must be submitted to ClinVar in addition to being put into the paper. Then you can ensure that the nomenclature is correct, which has never been an accurate thing in a lot of papers. But also annotations could be structured on those variants in parallel with talking about them in text in a paper. So in that way, we could start to make better use of databases like ClinVar with, over time, more structured fields of evidence than is there today, quite honestly. And that the paper is really just a thoughtful summary of information that's primarily stored elsewhere as a compendium to the paper. And that's not just true in genetics. That's true in probably all domains of scientific inquiry and analysis. So that's my hope, that we are increasingly structuring our evidence at the start and sharing it ways that primary evidence base is shared as structured form, not just summaries of it in manuscripts.
Yeah, I would second that too. Particularly having done a lot of curation, I think the things that are particularly difficult of some of the really old papers, and you just have a protein nomenclature and you're sitting there like, how do I even add this to an interface, because there's just a protein change. So I'm hoping that some of these large curation efforts can help to parse through that. They're quite painful right now, and we can hope that in the future things will be a lot more standardized. But we've even talked about using public annotation tools, such as Hypothesis is one of the annotation tools we've explored a little bit, where efforts like ClinGen can go and publicly annotate a paper and have little comments like nomenclature here, segregations here. So it might be a little bit difficult for machine learning to pick that up, but at least it's a little bit more digestible than just the lack of standardization that a lot of these old papers have, which is particularly difficult for gene curation, where you want to see historical papers.
Just wanted to second Heidi's point about, to some extent, NLP is a very sophisticated solution to a problem that is a horrible problem that is largely of our own making, in the sense that we put all the data in this way. And it's horrible for all the reasons Heidi pointed out. And the other horrible component of it is that we as a community have allowed so much of that data to be locked behind paywalls that make it very difficult to do systematic analysis across journal articles. We have let this data be concentrated by a series, a set of very large for-profit corporations that hold that data away, and that is the other big challenge for us, I think, is getting access to that data. Part of me just wonders, one of the reasons I like thinking about Mel's question about in 5 years' time is that I suspect, over the next 5 years, we will have started to curate a lot of that literature in the right way, and we will have re-observed, as we start sequencing more and more patients, a lot of the mutations that are currently present in the literature, and it will become increasingly less important that we go back and try to tease out painfully all of that unstructured information from the literature, and we'll instead be able to rely more and more heavily on these structured databases, and there I think the problem becomes much more tractable.
So do you think if labs keep sharing data with ClinVar with more evidence and summaries, do you think for the concordant class, using the concordant classifications as your positive controls, machines can be trained to basically know to pick up what to pick up for classifications like from papers, because they're trained on concordant classifications with the evidence. So the machine is that what you're trying to say? Essentially sharing more data to train
more structured data and
more
robust
structured
data.
yeah.
Variant interpretation at its heart is a classification problem based on very complex sets of parameters, and that's the sort of problem that lends itself very well to machine learning approaches. The problem up until now is that there just hasn't been sufficiently large sets of data that are both sufficiently well structured and sufficiently accurate that you can actually get these systems to work appropriately. But given a large enough ClinVar with really heavily structured evidence that goes into it for a set of variants, then variant interpretation does become more and more of a machine learning problem. It's never going to steer entirely away from needing a human component, but I think it becomes a very powerful decision support tool on top of the human piece.
Do you have a vision on how long will that take?
It's all going to happen in 5 years. 5 years. Everything changes in 5 years. This course in 5 years, we'll be talking about interpreting the results of machine learning algorithms. No, not quite. But I think that's the sort of time frame where we will see. I mean, a lot will depend on the growth of ClinVar. I think the machine learning methods are becoming sufficiently sophisticated now. It's really so much is dependent on the quality of the underlying data.
And I think we've had great success with ClinVar of labs submitting their classified variants, and the issue sort of I think that'll hopefully be rectified soon is the sharing of case level data. And it's very easy or easier for labs right now to say, yes, here's the variant, here's my classification. But when it comes to, can you send me your evidence? Multiple labs will send me their data, and it's all in different fields. Some labs don't even store if these two individuals were from the same family or what other variants are in there. And so that is the big issue of how to share that type of data. And I think we need better standardization for labs of how to store their case level data, so that then there could be a better system to share each other's case level data and be able to look at that and get the information out without just sending free text summaries or random information about each case in an unstructured way.
So first I would say phenopackets. So for those of you who want to coming to a theater near you. But that was the whole goal of phenopackets, was actually to share case level information. It came out of some of those early labs discussions, and also with journals. So I think both Heidi and Daniel about how we have to rethink the way we just tell. The literature started like 350 years ago with the first philosophical transactions, was about telling stories about science. And it hasn't actually changed since then. And so I think the other thing about that is that we actually, those tables that go in every case report, like, they're so infuriating because you don't know which phenotypes went together in one patient, right? And so that's what these phenopackets efforts are trying to do. And so I think getting the journals to really upfront require structured case level information in the same way that we see in databases is definitely a key thing. So really pushing on the journals to do that. And then once we do have those data, we can actually train our algorithms better. I think the second thing that I wanted to point out too is that what's also going away is the literature as the sole source of our academic integrity, and and that we're going to start and there's some efforts to really promote attribution and provenance of contributions to public databases and other kinds of contributions that are not a journal article, right? That fundamentally how we value our contributions as scientists is not going to be valued only on journal articles anymore. And so it's interesting, I was at a rare disease community, and I stood up at the end and was so surprised that they said there would be no photographs, no data sharing, no tweeting, no nothing. And I was like, why are you guys not announcing to the whole world that you have these needs and you want people to analyze your cases? You're so few of you, And one of the more senior scientists stood up and said, because if we do that, our postdocs aren't going to be able to get that Cell or Nature paper, and therefore they're not going to be able to get a good job. And I said, well, don't hire anybody who doesn't share their data. Your job descriptions should have these things. Start valuing the things that are not the traditional Cell or Nature paper. So I think, I know it's a long way from the question about machine learning, but but this is the kind of, we have to think about machine learning in the context of the whole ecosystem of resources that we have out there. The literature is just one of those resources.
And
go ahead.
I'd say even from a case level data, only a very small % of all the cases we've ever seen in the clinical lab go into a publication. So there is that huge other source of data of all of our unpublished cases.
Yeah, I was just working on a grant over the weekend, and I was trying to document my lab's experience with variant classification as evidence that I'm prepared to, in this case, sequence 1 million genomes and interpret that for the All of Us program. But I could write, my lab has interpreted over 22,234 variants because we've submitted them into ClinVar, and it's public record, and I put the URL where anybody can go and see that. So, like, there's no publication or anything out there that could document my experience better than my public submission to ClinVar. So I completely agree with that. You know, in some ways, sharing information in a public database is a absolutely transparent, unequivocal way to say, I have experience doing this, and I've shown that I've done that because it's right there and I gave it to everybody. So there are, you know, some concrete benefits that people can, you know, have from doing that. Anyway, another question?
Put that URL on your CV and your NIH BioSketch, because we've changed the BioSketch standards. You can put it there, especially for the people in your lab, right?
That's right.
So as a genetic counselor working on variant interpretation, I definitely appreciate the value of data sharing and ClinVar and things like that. But at my previous job, I was at a very large public hospital in Chicago with a lot of underserved populations, a lot of patients whose families weren't in this country, who they didn't have contact with their family members. A lot of my patients didn't even have cell phones or internet service. And as soon as I brought up data sharing, a lot of them wouldn't sign the consent form and would say they didn't want genetic testing. So now you have patients with possibly hereditary cancer syndromes who aren't getting genetic testing because they don't have a choice to opt out and still receive genetic testing at some labs. So I don't really know where I'm going with this, but I guess how do we tackle, I think it's easy for all of us here to appreciate the value of data sharing, but how do we communicate that to other communities who have been taken advantage of in the past and who are very vulnerable when it comes to sharing healthcare information?
Yeah, totally understand your point. And I think that goes to, we just need to do a better job of educating those people what exactly it is that we're going to be sharing. So Heidi mentioned our one-page consent form that has some associated videos with it. And as part of building that, we did several focus groups, including focus groups with patients, to just say, you know, what do you even think of this idea of broad genomic data sharing? You know, is this something that's acceptable to you, or, you know, what do you think about this? And a lot of I mean, they've never considered such a thing before, so they don't even have a place to start to know what we're talking about. And for a lot of them, especially when we showed them what ClinVar was and what kind of information we were sharing, they were immediately like, oh, it's just that? That's what you want to share? Okay. You know, I'm thinking you're going to share my name, my address, you know, a picture of me or something. And when they actually see what it is, you know, in this case, you know, a variant and its interpretation, they felt more at ease. And so we did try to incorporate some of that into those videos, which of course anyone's welcome to use in their setting. But I think just, you know, and a lot of times you don't have time to do this necessarily, but when possible, to just explain, you know, it's not that bad thing that you might be thinking of in your mind, like a photograph or what have you. It's actually this, and now that you know this, do you feel differently? But of course, we never want to make people feel like they're forced to share if they don't want to share.
I do think that is very hard to do with individuals who don't have a lot of trust for medical professionals from the beginning and who can't access those resources on the internet.
So we do have a brochure. We have a printable brochure for the video as well, just say that. So we did think about the people who don't have access to the internet or don't want to access the internet. That was another point that we got. People said, I don't, especially older individuals, I really don't feel like sitting there and pulling up YouTube and watching a video. So we do have it printed. We have it translated into several languages. But no, I mean, it's a fair point, and I think that's where you just have to have that, you know, that personal conversation and build up that trust with your own relationship with them before you ask them to do anything, not just the data sharing.
I mean, but also acknowledging there are like perfectly sound reasons for mistrust in a lot of these communities, and it's hard to get those things back, obviously, and particularly in the context of the American healthcare system, which has mistreated, you know, people in many different ways across many different contexts as well. So that's going to be hard. Education will get some of the way there. Greater penalties for misuse of the data, I think, will, you know, help to some extent as well. And then, I don't know, at some point we're going to have to fix the American healthcare system so that it treats underrepresented populations slightly better. That's obviously not going to happen for the next few years, but, you know, at some point maybe we can start making some progress there as well.
I also think it's important to have trusted representatives of those communities who can sort of act as liaisons, so that, you know, there's not some, you know, white male, excuse myself, Daniel, showing up to an underrepresented minority population to tell them that they should do something. If they have someone from their own community who's been through and seen the world through their same eyes, they're much more likely to trust the advice from that individual than from someone who's not like them. So I think trying to engage, people who can act as liaisons to the communities that are underserved and underrepresented, I think is a critical way to develop that trust and engage that community.
I just want to point out we have 5 more minutes. So I think we have time for either one long question or 2 short questions, but I think with our crew that might be one. In a good way.
I guess, I think I work in the clinical setting, and today it's been amazing to see like the breadth of the tools that actually exist that are primarily built for the research setting. And I know that I've worked at 2 separate hospitals, we're sitting on these massive silos of clinical data, but we don't have any non-manual way of sharing that with researchers and with large things like ClinGen and ClinVar. And, I work at a well-resourced hospital now, but I didn't previously, and there weren't the resources. We saw 20, 25 patients a week. There were not the resources to even put that information in while we would love to. We couldn't even have, mailed you our charts because there wasn't the money to do that. So I wonder, is there some discussion around creating tools that are less manual, or creating relationships or jobs or funding for helping us as clinicians get you the data that you need for a lot of this curation, rather than these 2 worlds really not having a good bridge?
Yeah, it's a tough question, especially as we think about retrospective data collection. At some level, we have to accept that we may not get all the data that's from the world behind us. I watch Partners Healthcare invest $1.5 billion in switching to Epic, right? And of course, if you ask any clinician, they just complain completely about Epic. But at the end of the day, the reason the decision was made is around accountable care, ability to interoperate and share data. And as much as Epic has its immense limitations, it is far better than the homegrown systems that every hospital had until more recently. And so while I don't believe Epic is the be-all and end-all, it is an incremental step towards standardization and collecting, more electronic data that can be shared in easier ways than we could before. And so I think we have to think about it in not how am I going to go get, 1,500 pedigrees out of progeny, like we were talking about before, that I've collected over the last 10 years, to when the patient comes in tomorrow, a brand new patient, what could I offer that patient that's different than how I've been doing things? And think about forward. And then, if we can solve that problem going forward, we can start to think about resources depending on the value of that old data, which sometimes maybe cut your losses and move forward. So I think we have to balance the value of that data with the resources it takes to get stuff out of old systems. But I think right now we have to have some focus on going forward and doing it better in the future. At least that's my own opinion. So there's an international effort called FHIR, the Fast Healthcare Interoperability, they're really working on data model interoperability between different EHR systems. And it's been a long road, and I think there was a lot of skepticism early on, but now there's quite a suite of tools that can be developed on top of that, and all the major EHRs are now working with FHIR to greater or lesser successes. But I think that's going to meet this prospective need to sort of start to build those kinds of tools in. Part of the problem with Epic, for example, is somebody at some point, some number of years ago, did an assessment where they looked at how many fields were in common across all the different Epic installations in the US. There were only 30% in common. So Epic has made their business on customization, but yet every hospital now is asking for interoperability. And so, with FHIR now actually mature enough, I think there's a real opportunity to have these suites of tools that can actually prospectively collect the data that we need for that sort of thing. And the examples that we heard earlier about being able to build an algorithm that can link patients together using things that it finds in the EHR record. Now we can build a tool that using FHIR that can actually work prospectively to collect the right information moving forward. So
I'll put in one final plug for Genome Connect. seriously, like, get the patients to help you. Do you know what I mean? Like, every single one that you do a test on, make sure they walk out of your office with a copy of their test result, which they should have anyway, and information on how to sign up themselves and submit it themselves. You know, I mean, we didn't talk about it in detail, but we asked the patients fill out their own health survey. They tell us about their own health. Then we have them upload a copy of their report, and our staff pulls the variant information out so they don't have to do that potentially more complicated part. And then we submit all those to ClinVar. So, you know, leverage the resources that we have from our research grant to help you get some of that clinical data in without even your manpower, you know, engaging those patients as partners too, while we wait for some of these more elaborate solutions to come about.
All right, well, thank you again to all of our speakers. So we do have just some final housekeeping, because I know you guys are going to miss my housekeeping from the last 2 weeks, or 2 days, 2 weeks. It feels longer than that. So I do want to announce, I have found that this course was successful. I hope you all have too. A family was actually diagnosed by one of our people learning. So Jen Partlow went from a somewhat newbie and seeker to actually diagnosing a family. And so we are very grateful for that. So thank you. New connection, yeah. I don't have a trophy for her. I felt like I should have, but I didn't get one, sorry. So new connections were made. Please stay in touch. Feel free to email me if there's any questions you have, any feedback, again with the survey, but please feel free to just submit that to me directly if you feel you want to. And then I also heard that there were many Hamilton tickets bought during myself and Heidi's talks this morning, even by Heidi herself. So to all of you going to Hamilton, congratulations. I'm jealous. Enjoy. And I will upload all of these, so I won't go through everything, but for any of you who are interested in submitting cohorts or cases to our Center for Mendelian Genomics, we do have a few requirements. These are all located on our website, which is where the course website was located. And then I also, I found this new tiny URL application, so I'm making it for everything. So we have a tiny URL for Broad CMG cohort application. It takes you to our online submission form. Again, you can also email me if you have any questions about that. I also wanted to plug CMGs in general are all about education to the community. So one of our brother, sister, cousin CMGs at University of Washington, who we work closely with, offers a 1-week-long course that is very much focused on bioinformatics and analysis. It's a very intensive course. So if you really enjoyed the seeker part of this and want to just dive even deeper over a week, they do have a course coming up in August. It's the 13th through the 17th. They offer this every year, so I know this could be a bit aggressive to like come to this and then go to that, but we wanted to plug it. Also, there you can contact Colleen Davis, and there's flyers out on the table that we have out front, and you can grab one of those to find out more information. So with that, I will just say thank you to everybody for coming. We really enjoyed this course, and we hope to see you all in future years. Daniel did promise it's coming in 5 years.
I just, Daniel and I, on behalf of the CMG and ClinGen group, would like to say thank you to all of the team members who worked to put cases together and workshops and sat at the registration desk and made websites and everything. But a particular call out to Danielle, can't reach back there, and Sam. I have personal observation of the amount of work that's gone into this course with the number of communications, so thank you very much. We appreciate it.